All right, everyone. Thanks for joining us on our webinar. I'm just going to get started. My name is Leah, and I'm the Scientific Knowledge Manager at CellCAN. So just before Dr. Viswanathan starts, I'm going to tell you a bit about CellCAN and what we are up to. So CellCAN was incorporated in 2014. We had funding from the Networks of Centers of Excellence. And our vision is that the cell and gene therapy manufacturing in Canada can operate under a common seal of quality. And our mission is to improve the quality, safety, and feasibility of cell and gene therapy in Canada through optimal manufacturing practices. We have 4 business pillars. The first is to build a strong Canadian network. And we'd like to do that by increasing best practices and creating a common seal of quality or common quality standards across the country. We want to increase collaboration and break down silos between different manufacturing centers. We also like to advance regulatory standards. We do this through partnerships with Health Canada, and one of the things we're working on now is regulatory modernization. So what we want to do is increase legitimate, authorized clinical trials while decreasing unproven fee-for-service clinics that are operating in the country. We also want to generate effective outreach. One of the ways that we're doing that is through systems like our webinars, and we also have training programs and partnerships with other groups. And so through all of this, we'd like to position Canada as a world leader in the cell and gene therapy. We do have another upcoming webinar, so if you haven't subscribed, be sure to subscribe to our list so you know what's coming up. Our next one is going to be by Sam Wadsworth from Aspect Biosystems, talking about bioprinting. We also, as CellCAN, are offering more and more training initiatives. So recently we did a hands-on GMP training workshop at the CETC in Montreal. There will be more editions, so you can sign up on our website for more information. We're also advancing regulatory standards in the community. If you would like to be part of our cell therapy stakeholders group, let us know and we can connect you. This is where we're communicating with Health Canada to get more information on advancing the regulatory standards in the country. Another thing that we're heading to is ISCT in Paris. We're going to have a Canadian pavilion, so if you're Canadian, feel free to stop by. If you're not Canadian, come and see what's going on in Canada. And our speaker for today, if you like this webinar, is also chairing a session at ISCT in Paris. So be sure to attend the MSc plenary session as well. We had a very successful strategic forum in 2019. It was called Time to Gown Up, and we will be holding another edition in 2021. That will be in Ottawa. So sign up. We'd love to have more speakers and sponsors. And with that, I will hand it over to Dr. Sowmya Viswanathan, and she's going to tell us about regulatory standards and the NSNRO. So Sowmya, if you can just...
Great, thanks everyone. I'm going to share the screen in a second.
Great. One more thing, housekeeping.
Can you guys hear me?
Yep, I can hear you. If you have any questions, just type them into the chat box and I'll ask them at the end. That's all I've got. So over to you, Sowmya.
Okay, thanks, Leah, and thanks to CellCAN for setting up these really informative webinars. And I'm going to talk to you a little bit about this new substance notification regulation and how that applies to the audience who are mainly involved within cell and gene therapy and gene-modified cell therapies. So in Canada, as you all know, the cells, genes, and are covered, or gene therapies are covered as drugs, and they're regulated under the Food and Drug Act and largely under the Food and Drug Regulations, although other regulations like medical device regulations or cells, tissues, organs regulations may apply. In 1999, Environment Canada, now changed to Environment and Climate Change Canada, enacted what's called the Canadian Environmental Protection Act. And in 2005, 2 regulations were created under this act. This was the New Substance Notification Regulation for new organisms and a New Substance Notification Regulation for chemicals and polymers. The idea was to be able to assess really the environmental and impact on human health for any toxins that are released into the environment under these regulations. But as you'll see throughout this webinar, the original definition of living organisms under CEPA, which is the Canadian Environmental Protection Act, in 1999 is very broad because it essentially defines living organisms as an animate product of biotechnology. So viral vectors, genetically modified cells, and actually even cells, whether they're autologous or allogeneic and are manufactured and processed by quote-unquote biotechnology, are captured under this very broad definition and therefore are likely to be subjected to these regulations. So for this talk, I'm going to really focus on organisms rather than the chemicals and polymers section because that's what this audience is really interested in. So as I said, the goal was really to ensure that no new organism, and especially microorganisms, were introduced before the risk had been assessed. But because the net was so very broad and vague in terms of the definitions, living organisms, microorganisms, and organisms other than microorganisms are all being captured here and maybe increasing some of the regulatory oversight and burden, which we will discuss. So as I said, a living organism is defined as an animate product of biotechnology. And within that, there is also definitions for microorganisms under CEPA. And this includes bacteria, fungi, archaea, algae, but it also includes virus and virus-like particles and subparticles, even though these are largely replication incompetent, and most virologists do not consider viruses to be living organisms as they're not able to replicate on their own. Nevertheless, they meet the definition of a microorganism under the Act and are therefore captured under living organisms. Very interestingly, they've also included culture of cell of any organism or any culture other than a pure culture to be included in this definition of microorganism. So as a result, really all kinds of vectors, oncolytic viruses, cells, gene-modified cells, which are used in a hospital or a clinical setting outside of a contained facility are captured under the New Substance Notification Regulations and therefore need to be assessed under these regulations. So this includes things like CAR T cells. It actually includes any culture expanded cells. It includes the vectors themselves, which are also considered new substances. And these could be replication incompetent vectors, or in the case of viruses, the replication competent ones, even if the trophism is not human, are still captured under these regulations, and therefore will need to be compliant with the new substance notification package. So currently, anything that is not on the DSL, the domestic substance list, will trigger the requirement to notify the authorities that you have a new substance. So for chemicals and polymers, this is anything over 100 kilograms per year, but you can see for organisms, it's basically triggered immediately. And it can be triggered at the time of importing, at the time of manufacturing, at the time of submitting a clinical trial application, or at the time of a new drug submission for market authorization, or interestingly, if the substance is on the DSL, but the activity that you're now proposing is significantly different from the original activity which required it to be on the DSL, again, you have to submit a new notification to Health Canada and to Environment and Climate Change Canada. But there are some exemptions. So the most important and obvious one is really a research exemption, which means that you're doing everything in a contained laboratory, whether it's imported or it's manufactured, but manufactured under containment level 2, 3, and 4. So essentially what that means is that your lab is covered under the Canadian Biosafety Standards and Guidelines, and your institute will issue you a biosafety certification, which is regulated by the Human Pathogens and Toxins Act, and which includes provisions for how, for example, you would dispose your waste. So this is considered a contained facility and a contained release. So it's not a widespread release, and this means you're exempt. If it's not being used in a clinic, if it's being used, for example, in a clinical trial, in a hospital or an outpatient or any kind of clinical setting, it is not considered a contained facility. That means that does trigger it to be compliant with the notification process and requires you to go through the new substances notification process. Other ways that it can be exempted is if there are acts or regulations that are equivalent to CEPA. So there is a schedule in CEPA, which is the Canadian Environmental Protection Act, which basically states that if there's other acts that are equivalent in terms of environmental assessment to CEPA, then there's no need for redundancy. So there's no duplication. So Pest Control, Seeds Act, Fertilizer Act, Health of Animals Act, all of these are listed in Schedule 4. So if the living organism or microorganism is regulated by any of these acts, then it doesn't have to go through the notification process because there is an equivalent act to regulate it. You'll note that the Food and Drug Act or the Food and Drug Regulations is currently not included in Schedule 4. So anytime you're submitting something which is considered a drug or a medicinal ingredient for a drug for a clinical trial or market authorization, you do need to go through the new substance notification process. Some other exemptions include things like if the substance is in transit, if it's an impurity, or if the substance is already on the DSL. So what is a DSL? The DSL is this domestic substance list, and it's essentially an inventory of about 23 substances that are used in Canada. And I've provided a link to this DSL list on the Environment and Climate Change Canada website. And the first place for you to start would be at this list to search for your substance to see if it's already on the DSL. Now, it only gets put on the DSL if it is being manufactured or imported. So if there's a notification sent to Environment and Climate Change Canada that it's going to be imported or manufactured and they've reviewed it, then it'll be placed on the DSL. If there isn't an active intent to manufacture or import, then it may not be on the DSL. Additionally, many sponsors place their items on the DSL but request it to be masked for confidentiality reasons. So if you do a search, it may not pop up because it is masked. In such circumstances, you can request Environment and Climate Change Canada to perform essentially a confidential search for the masked item. And they'll do this within 28 days and get back to you. If your substance is still not on the domestic substance list, essentially it's considered new and it requires the environmental assessment prior to any manufacturing or importing or use. As I mentioned, there is a caveat. Sometimes it may be on the DSL and may be listed there for a different purpose than the one you're proposing, in which case it's still considered a new substance or a new significant activity, and they'll still require you to comply with the notification process. So you've searched the DSL, you've done a confidential mask search or requested one, you've not found the item on the DSL, so now what? So essentially what this means is that you need to complete the NSNR package and all the applicable schedules that go with it. There's a number of schedules, but for the purposes of this talk, I'm going to really focus on microorganisms, which include genes, viral vectors, bacterial vectors, and gene-modified cells. So this is really Schedule 1. Back in 2010, there was a guideline issued for completing these, the technical components of these schedules. And it's actually a very detailed guideline that you'll find for each of the different sections. And there are about 9 technical sections that need to be completed. We have requested, as part of our feedback to Environment and Climate Change Canada, to issue a new draft guideline, which they did circulate for comments, and it was supposed to be released in 2019, but it has not been formally released as yet, so stay tuned. Hopefully that should be coming, and we'll have more specific examples on how to fill out the Schedule 1 and the various technical sections. There's also an application form, which you can download from this link, and it has actually a really nice checklist that you can walk through to see what the different sections are and what you need to what you need to fill out in them. How you need to fill them out is still in the guidelines. But the most important thing I can give you from this webinar is to have this pre-notification consultation or a PNC meeting with Environment and Climate Change Canada to get guidance on how they want you to fill out the various technical sections, as their mandate is really to look at the environmental risks. So the types of information that they want you to put in this is a little bit different from, let's say, a clinical trial application or an NDS submission. So having that meeting with them is really the best guide to figure out what types of information you need to put in there. As I mentioned, there's a number of different schedules, and it really depends on where you're introducing your microorganism. But for the purposes of this webinar, I've gone with Schedule 1, which is basically release anywhere in Canada. And right now, at least formally, there's a 120-day review for Schedule 1, although I'll talk a little bit about this because they have streamlined this based on feedback that they received from the stakeholders. So I'm going to very briefly go through Schedule 1 and the types of information that is required, and you'll begin to understand how difficult and laborious this might be. So as I said, there's 9 technical sections along with the administrative section, and you are required to fill in various aspects, for example, the strain history. This is possible, obviously, and you can refer to the literature to get strain history, but let's say you have a lentivirus that you're looking at, it's a 3rd generation lentivirus, then you may have to cite literature deriving it all the way from the HIV original strain to the current modification of your lentiviral vector. So this could be, thousands or tens of thousands of literature searches. So how do you, how do you draw some reasonable boundaries around that? So the PNC meeting could be really useful to understand what they're looking for in terms of this assessment and what's a reasonable amount of literature, for example, to present just for this one section. Other aspects may be more difficult. So, for example, life cycle of a vector that is of a viral vector that's replication incompetent and only replicates essentially in a human host cell by hijacking the cellular metabolism of the host cell would be difficult. Other things like resistance to antibiotics, tolerance to metals, pesticides, these may not be applicable. So how do you begin to answer these various sections? Section 2 is all about importing and manufacturing information. So this is probably a section that you will have some redundancy in terms of in your CMC section in your CTA about how you're manufacturing, for example, your viral vector lots or how you're transducing your cells. And this information will be there in the CTA, but it's also captured here. But again, the intent of this is a little bit different. There will be lot-to-lot variability, and again, having that consultation meeting with Environment and Climate Change Canada is best to figure out how to fill out the various sections in this particular technical section 2. This section is all about how you're introducing your microorganism and how this is different from the natural habitat that you find the microorganism in. Again, very difficult to answer for your viral vectors, which are mainly where you're mainly introducing them into human cells or into humans during the course of a clinical trial. So they don't really have a natural habitat, so that comparison can be very difficult to figure out and to answer this. So again, having that discussion with them in terms of what you can and can't answer will be really important. Same with how they interact with other plants and other species in the environment. Again, they're not really designed to interact with these animals and species. But you can't simply put not applicable in many of these sections because they don't really like that. So they are looking for specific types of information that you can provide in here, which speaks to the inability of their viral vectors, for example, to interact with various other species in the environment. So again, getting that consultation is the most important aspect, get clarity, at least at this stage. The human health effects information, this is probably something that you are going to be designing as part of your clinical trial. So you will likely have shedding studies in there where you're going to be collecting samples, feces, urine, saliva, and the type of samples and the frequency at which you collect the samples and the duration for which you collect the samples are all determined by sort of the tropism of your vector, its infectivity, its pathogenicity, its immunogenicity. So these will be talks that you would have had with Health Canada while designing your clinical protocol to figure out the types of clinical shedding studies that you're going to be doing. Now, in most cases, there are guidelines, for example, that are put out by ICH and by the FDA that speak to sort of these, how to design these shedding studies. And this will feature very largely in your clinical trials. You may have also been required to do some shedding studies on your animals. For example, if you have a replication competent virus, an oncolytic virus, for example, then you may have been asked to collect data from your animal studies, and that may have been part of your animal study data package. But again, because of the difference in tropism of your vectors and the difference in immunogenicity between the animals and the humans, human data is always preferable, but you're only likely to collect this data towards the end of your clinical trials. So a lot of this information will not be answerable when you're first initiating your clinical trials. And many of the guidance documents from the FDA, for example, actually advocate collecting this in later phase clinical trials in phase 2 or phase 3, because they're cognizant that there may be manufacturing changes or tweaks, for example, to your vector or your manufacturing process, which may in turn affect and have consequences, for example, in terms of replication competence or infection or shedding. So they require you to do many of these studies later on in your clinical trials, or if you've done them early on, you may have to repeat them later on as you get towards your final version of your viral vectors or your gene-modified cell product. So all this to say that these different sections of Schedule 1 are quite onerous, laborious, and difficult to fill out, probably with the type of information that you will have at the beginning of your clinical trial. There is an opportunity, and CEPA does allow you to request a formal waiver from some of these sections if it meets one of these 3 criteria. So if it's really obvious that the living organism is not capable of being toxic, is either not toxic or is not capable of being toxic, and you can provide sort of evidence to support this, a waiver may be granted. Or if you're able to show that you can contain the living organism at all times during your handling and processing of it so that there's no kind of release to the environment or any impact on human health, then you can get a waiver. Or if you can really substantially prove that it's absolutely impossible and not really feasible to generate the type of data that you need to get that information, then you may be able to get a waiver as well. But be warned that whenever you put in a request for a waiver, it may or may not be granted. And if it's not granted, then that section that you have not completed is incomplete, and your package will not be reviewed until it's complete. So until your NSNR package is complete, the review clock does not begin on it. So sometimes granting these waivers and not getting the waivers granted may actually delay your review process. So I'll just talk a little bit about the review process. So when you're submitting your clinical trial application, typically to the Biologics, Genetics, and Therapeutics Directorate, that's a 30-day review that they take to look at the clinical and quality aspects. The NSNR is a 120-day formal review by the Environmental Assessment Unit, which is kind of a liaison group between Health Canada and Environment and Climate Change Canada. But they don't really have access to your clinical trial package. And you can submit the NSNR package either prior to or concurrently with your clinical trial application package, but beware that they have no visibility to the CTA, just as the BGTD group doesn't really have visibility to the NSNR packet. So these are 2 completely different packages that are reviewed by 2 different groups. Recently, the Environmental Assessment Unit group has committed to shortening their 120-day review to a 30-day review. This is an informal commitment so that they're more aligned with the CTA review process. And this is something that they have been doing as part of the feedback they've been receiving from stakeholders, including CellCAN and many of the biotechs industry in Canada that's been providing sort of feedback to make this process more aligned with other jurisdictions. But you can imagine that because of these two independent review pathways, there is delays because you have to prepare essentially two packages. There is redundancy in the types of information that you're going to be providing, especially related to the preclinical and clinical shedding studies and some of the manufacturing information. And it creates a lot of uncertainty in terms of whether your package is complete and how it's reviewed, because the two agencies or the two groups have slightly different mandates in terms of how they're assessing the review. And obviously it takes a lot more resources to get these two packages completed. And this is different from, for example, what the FDA does. They really have a single window for review, and it's essentially your IND applications. Your investigational new drug application contains the environmental review components, and it's included in it, and it's done essentially by the same group. And as I said, there's a lot of guidance documents available put out by the FDA, for example, on how to design some of these clinical studies for collecting shedding data from the various samples that you'll be collecting over the course of your clinical trial. The European Medicines Agency is a little bit more complicated, and they do require an environmental risk assessment to be completed. And like the FDA, this includes data on shedding, stability, pathogenicity, replication competence, susceptibility of the immune system, unintended transfer, and the consequences of such unintended transfer. Typically, this is part of market authorization, which is when the EMA really assesses your application. Clinical trials are typically done at the level of the national health authority. So the competent health authority for a given nation will be reviewing the clinical trial dossier. And here there's quite a bit of variability between countries. Some countries consider clinical trials to be deliberate release and therefore require sort of this environmental risk assessment package in full to be completed and assessed, while others consider it more of a contained release, and so they are taking more of a staged approach and do not require as much information to do the environmental risk assessment during the clinical trial stages. So there's quite a bit of variability depending on what country you are in Europe in terms of the types of information you're required to provide at the clinical trial stage. When you look at Japan, they also have an environmental review, but this is a joint review that's done by the Ministry of Environment and the Ministry of Labor, Health and Welfare. And they basically follow the Cartagena Protocol on living modified organisms, and they classified, at least for environmental review purposes, their products into sort of type 1 and type 2. And type 1 is really where many of the gene therapies and viral vectors fall, because it's considered that these are the kind that have no preventative measures against environmental dispersal and therefore require this type of joint review by the Ministry of Environment and MLHW. Type 2 is where they assume that there are containment measures in place while you're handling the living organism. And so it's a staged approach, and so they require less stringent environmental review at that time. But as I said, this is a little bit different from what we do here, because we have separate independent reviews to two different agencies who don't really have exposure to each other's dossiers, and this requires a lot of duplication of effort, delays, and uncertainties in the approval process, and it has been somewhat of a deterrent for companies and sponsors that are looking to do especially gene therapy trials in Canada, because it adds this additional review step with an additional group and additional uncertainties and delays. So we've been very concerned about having these sort of more non-streamlined review process compared to other jurisdictions. And so we've been giving this feedback to the government, and to their credit, they've been really listening and responding to the stakeholder concerns. So one of the first groups to lead this was CellCAN, along with Biotech Canada, which is really a conglomeration of various biotech industries in Canada doing a number of trials, including cell and gene therapy trials. So a workshop, which was an invitational workshop, was held with Health Canada and Environment and Climate Change Canada, and included other stakeholders like BioCanRx and C3I, and we actually laid out the position of how these additional environmental review process was creating important bottlenecks in enabling clinical trials, especially in the gene therapy space in Canada. And we've published this proceedings from this workshop in Frontiers in Medicine. It's an open access journal, so this report is freely available, and it really discusses sort of the background for the process for this notification and some of the recommendations that we made that came out of this workshop. So really about 5 recommendations were made to Health Canada and to Environment and Climate Change Canada. And sort of the most obvious one was to include the Food and Drug Act, the FDA, under the list of exempted acts in CEPA. So to really put it in Schedule 4, just like the other Pests Acts and the Fertilizers Act and the Seeds Act, we could put the Food and Drug Act and have the Food and Drug Act under its auspices and under its regulations do the environmental review that CEPA is supposed to be doing. This is obviously a longer term plan to bring about such an amendment to an act, which would take quite some time. Some more intermediate changes were considered more policy changes, which would be more interpretive changes to the act. So for example, clarifying the definition of living organisms so that something like human cells touched by biotechnology for use in human medicinal products could be exempted from that very, very large definition of what a living organism is. And that might immediately exempt at least autologous or allogeneic cells, for example, that are not genetically modified from being previewed by CEPA's environmental risk assessment. Another sort of policy change that we proposed was maybe to narrow the scope of microorganisms so that it doesn't include virus-like particles or subviral particles, which are really replication incompetent and are therefore not really considered living and should not fall under the definition of living organisms. So by narrowing that scope, we thought that some of the viral vectors that are not replication competent could be excluded and would not be subject to the new substance notification requirements. Another idea that we proposed was really to defer the full review of the NSNR till the market authorization stage. So to remove it from the clinical trial application stage and to have it be prevalent at the market authorization stage, because by then you would have collected the types of clinical data and the clinical shedding studies that you're required to do anyway as part of your clinical trials, and you would have the information where you could talk about the impact on human health and be able to sort of more competently fill out some of the sections of Schedule 1, which are really at a little bit of a disadvantage to do at the beginning of your trial, because you may have some animal data and you may have some literature data, but it's not really relevant to your particular viral vector or your particular gene-modified cell product. And you probably don't have human data on it. So at the end of the clinical trials, you probably have a much bigger data set to be able to tap into to more thoroughly fill out that assessment. And then the last one we asked was to give us some more practical guidelines with concrete examples to fill out Schedule 1, especially for people who are doing clinical trial applications. And that's something that they have acted on. As I said, that guideline is to be issued hopefully shortly in 2020. So as I said, the government has actually been really responsive to the stakeholders' concerns, and they've been doing a number of, they've taken a number of initiatives. So one of them has been extensive consultations that they've held with stakeholders, led somewhat by the Treasury Board as part of that initiative. And they've sent out these cost-benefit analysis surveys to really gauge the lost opportunity cause of having these additional regulatory burdens and how this is impacting industry and sponsors from doing clinical trials or bringing these cell and gene therapy products to market in Canada. So they've been very, very receptive to receiving this input from stakeholders. And they've actually put a report back out to the stakeholders in 2019, where, again, they're interested in making some regulatory amendments, including bringing this under the Food and Drug Regulation, which is actually one of the requirements that we asked, and to be very specific in terms of having data requirements for medicinal ingredients in drugs, and aligning the notification process with the drug review process, which is something that they've already done informally, as I said. And the Environmental Assessment Unit has committed informally to having a 30-day review rather than the longer 120-day review, so that they're much more aligned with the CTA process, even though it's still independent. And then also changing sort of the data requirements where possible so that there's more alignment with what's being done by the FDA and the EMA. They still want to have sort of 3 stages of notification. So again, the import or the manufacturing stage, the clinical trial stage, and the market authorization stage. And either, you would still be required to submit the Schedule 1 of the new substance notification regulation, and you would still need to fill in all of these technical sections so that requirement is still there. But they introduced, or they're proposing to introduce this new concept of a conditional exemption, where they're saying that if the increase in concentration in the Canadian environment is very unlikely, then perhaps you could qualify for a conditional exemption. So you would still need to provide rationale that there is no change in environmental concentration, there is no replication in the environment that's possible, or that it's really extensively biodegraded or rapidly metabolized, so there's no real widespread environmental release. And if you do that during your CTA or your market authorization stage, you could be qualified for a conditional exemption. When we specifically asked if this could be granted to non-replicating lentiviruses using a cell or gene therapy, the answer was that yes, it could, but again, they're going to do this on a case-by-case basis, and this would probably be done through the pre-notification consultation meeting, where it could be decided that this would qualify for a conditional exemption. You would provide, again, the written rationale along with the supporting data as outlined in your pre-submission meeting, and it could qualify for that conditional exemption. They also talked a little bit about the list, which was basically to create a list of exemptions, the things that were conditionally exempt. So it's kind of like the domestic substance list, but it would in fact be one of medicinal ingredients that were either fully assessed or that were conditionally exempted. But what's not clear, and I think they're still looking to figure this out with the stakeholders, is how specific the level of information will be for the medicinal ingredients. So for example, if you're getting a lentiviral vector put in there, does that mean that all lentiviral vectors are captured? Or if you continue to tweak them, will each one be considered a new substance? So where does that, how broad does that have to be on the list? Because once it's on the list, then you can go back and say, okay, it's on the list. I'm not going to keep doing the notification process because it's already on the list. So this is something that they're still really open to and looking to get stakeholder input to in terms of figuring out how specific that medicinal ingredient level has to be to sit on the list. Because again, I said once it's added to the list, that means you no longer need to notify it as you use it in different stages, whether it's in your import manufacturing, your clinical trial application, or your market authorization stage. But be aware that these are all proposed reforms, so this will have to go through the full regulatory amendment process, which can take up to 2 years. So this is not immediate. So in the immediate timeline, you're still required to submit the new substance notification regulation, but there is light at the end of the tunnel because they are looking at making some of these regulatory reforms. E2I, which is really the Environmental Assessment Unit as part of E2I, has also been looking at how adding the Food and Drug Act and the Food and Drug Regulation to Schedule 4 of CEPA, because then everything would just fall under the Food and Drug Act and the Food and Drug Regulations. But they need to get a legal opinion, essentially, from Justice Canada to be able to declare that legally they're equivalent in terms of environmental regulation. But you could imagine if that were to take place and everything would fall under a single review and would not require sort of these two different submissions that would need to take place. And it would still be a staged approach from importation, clinical trial authorization to market authorization, which is consistent with the current regulatory reform that they're proposing anyway. So the last little bit I wanted to touch with you is some of the initiatives that CellCAN has going in this area. So they've been working very well with Health Canada and the Health Products Food Branch to provide feedback on these various government-led initiatives, participating in these stakeholder consultations, providing information for these business surveys, and with partners like Biotech Canada, who really are representing many of the biotech industries in Canada. We've been engaging and sort of keeping the pressure on Health Canada and Environment and Climate Change Canada to continue with their reform initiatives. Another thing that CellCAN has been considering with the BC Cancer Immunotherapy Program is really to develop this test case, because the BC Cancer Immunotherapy Program recently launched their CLIC 1901 clinical trial, and they had to go through this new substance notification process, and they successfully placed their CAR-T product on the DSL as a masked substance. But they can provide us with some concrete examples of how they went about filling those 9 sections of the Schedule 1, so we could develop really a step-by-step guide with concrete examples of the types of information that we would need to put in. And I think this would be a really useful initiative that would be led by CellCAN and the BC Cancer Immunotherapy Program to put out this information, because while the other regulatory reform is taking place, is still required to comply with the new substance notification, and it can be challenging, as I pointed out, to fill out these various sections. And with that, I think I'll stop to acknowledge Dr. Tanya Bubella, who, I see that Fraser is spelled wrong, but pardon the typo there, but she was actually one of the first to sound the alarm on this this regulations and kind of helped us provide feedback through the cell therapy stakeholder group to Health Canada. And then when we partnered with Biotech Canada and Ron Bach, we were sort of able to mobilize and get a lot more groundswell support to have the workshop that we did, which was sponsored by CellCAN as well. And we've seen some really good progress in terms of policy initiatives that have been taken on and led largely, I would say, by the Treasury Board, but Health Canada and Environment and Climate Change Canada have also been good partners in some of these reform initiatives. So thank you very much for your attention, and I will stop sharing my screen so we can go back to questions now.
Great, thank you so much. We've got, just a reminder, everyone, you can throw your questions in the chat box. Sowmya, we have a question from Sarah Elizabeth Siegler. She says, the FDA typically grants the EA exemption for CAR T cells. Does ECCC or Health Canada plan to grant a category-wide EA exemption for CAR T cells, given the number of CAR T cell IND and IMPs under development?
Yeah, that's a really good question, Sarah, and that's kind of what we were hoping we would get with this conditional exemption process, to say that, sort of all lentiviral lentiviral modified CAR Ts could be sort of conditionally exempted, since they sort of have the same, we hope, types of environmental risk. But what we've heard from them is that, at least for now, that would be on a case-by-case basis, but the conditional exemption is not up and running as of yet. So as of now, you're still required to do the NSNR submission, as I understand it. But be aware that there is the CLIC 19 product is on the domestic substance list, so maybe there is opportunities to piggyback off of that, which is what we're hoping to do with our test case, and maybe reduce the regulatory burden. But as I understand it right now, there is no sort of blanket exemption that would be given. It's still going to be done on a case-by-case basis, and each time a person has sort of a CAR T, they're going to look at it and assess it and understand the environmental risk. And as I know it, so far it's only 2 that have been that are there. So one is the Novartis one, and the other one is the BC Cancer groups one that is on the domestic substance list. And I'm not even sure if the Novartis one is on the domestic substance list as yet.
Very interesting. And so Sowmya, you also mentioned that you could apply for a waiver, but that if you apply for the waiver at the same time, then you could actually end up having to take things longer. So would you apply for a waiver before you've submitted the rest of the Schedule 1 form at all, or would you just try to take care of all of that in the pre-notification consultation?
So I think the pre-notification consultation is the best place to figure out what waivers you can submit. And again, as I understand it, the waivers are for specific sections of your Schedule 1. So it's not a waiver for the entire Schedule 1, but there may be specific sections that you can say, look, we don't have data on, ecological or environmental assessment impacts, and we're not likely to be able to generate this data until clinical trial is completed, and even then we're not going to have access to this data. So they may give you an indication of, yes, that's fine, and make sure you have this justification. So I think then you'd be okay to go forward with that. But that's a strategy that you need to discuss with them at the PMC.
Right. It sounds like it gets really complicated really quickly. And you've been through this process yourself, right? So could you tell us a little bit about maybe what you found to be the most difficult part of submitting the Schedule 1, or or something that went really easily that you found?
Actually, it's interesting, but I've not done the NSNR. Although we've done a lot of clinical trials, even with sort of gene-modified clinical trials, and it seems like the trigger was uneven, and Health Canada has become, let's say, more sensitive to the fact that it has been unevenly applied, with maybe more industry sponsors being flagged rather than sort of investigator-initiated trials. So I think they're now a little bit more strict about how they flag this for everyone. But even though we would have fallen under the definition of a living organism as an animate product of biotechnology, whenever we did our cell and gene therapy trials, we did not actually submit an NSNR. But with the recent wave of the CAR T trials and with the Novartis product being approved and their I would say, long time in getting the NSNR. So they got their DIN and they got their drug approved by Health Canada long before they got their NSNR. So it took them quite a while to figure out how to figure out the Schedule 1, and we were a little bit privy to that process because of their membership in Biotech Canada, and so they were very vocal about sharing some of those concerns as part of the stakeholder workshops that we've had. And so, behind them, all of the sort of the CAR T products that have been coming have been flagged for NSNR review, and I suspect that will continue with other gene-modified products as well. But we somehow got away with it, if that's the right term, but I think it's because again, there is a little bit of a low risk for non-gene modified cells. But if you're following the very strict definitions of CIPA, then yes, it should have been caught.
Okay. And is that, do you think that's because for your application it was just the gene modification, or because the cells were staying more in one place and they weren't sort of a liquid therapy, or?
So I think in our case, one, it was an investigator-initiated trial. So
Right.
I think there's a little bit less concern, and they tended to be more phase 1, phase 2 type trials rather than later phase trials. That was one.
Okay.
We haven't done lentiviral trials, although we've done sort of electroporation gene modification, but those were considered sort of very transient ones. So they're not really, and so maybe for that reason they were not flagged. Because they're transient genetic modifications. That could have been part of the reason, but it's difficult to understand why it has been applied unequally, with some sponsors getting flagged for it and others not. I know that a number of people doing just strictly gene therapy trials with, say, adeno-associated viruses, etc., were flagged for this. So they're not using cells at all, but just a straight gene therapy trial. So they were flagged and asked to go through the new substance verification process.
Okay. And do you have any recommendations on planning for the delays if you're trying to set up, I mean, like you said with, I think it was Novartis, if they have the IND equivalent submitted, but they are waiting and waiting on this,
Yeah.
how would you recommend people plan for that in the timing of their trials?
So I think when Novartis submitted it, they were still kind of following that 120-day review, which is exceedingly long. So I think the fact that they reduced that to 30 days is great. But I think starting early, so when you're doing your pre-CTA, you can do your PNC meeting at the same time. So you can, if you're in Ottawa, you can coordinate and meet with the EAU folks at the same time, or they're fine to do it over the phone. So I would say your pre-CTA you should also consider a PNC to happen either concurrently or around that same time, so you can plan your CTA package along with your NSNR package. As I said, there is some duplication, so it'd be great to figure out what parts you can get away with doing a copy and paste, and what parts you actually might need additional data or additional justification. You're probably not going to go generate animal data at this stage, but you may be able to do literature searches or come up with rationales that satisfy kind of the environmental aspect. So they're really looking at it from a containment and a release perspective, and less about sort of safety and efficacy, which is more the quality review that BGTV does. So understanding that they have different mandates and how you can present that information to align with their mandates is, I think, really important. So I would start planning for that around the time that you're doing your pre-CTA package. And again, you do have to prepare a PNC package for them, because they can't really review it unless they have a package as well. So it is another package that you have to prepare. And as far as I know, there's no sort of guidelines on what that consists of.
Great.
This is why we want to do a test, so we can publicly share some of this information.
Yeah, it would be great. So if anyone has any other questions, please throw them in the chat. I got a question about sharing the webinar. Our webinars will be posted on our website. I shared the link to some of our previous webinars, and this should be posted in about a month. Sowmya, thank you so much for giving us the time and sharing all this info. Really interesting to hear some of the difficulties that are happening right now, and also very interesting to hear about the regulatory reform that's happening moving forward and trying to make Canada a better ecosystem for cell and gene therapies.
Thanks, Leah. Thanks for moderating this. Appreciate it.
And with that, I think we will sign off. Thank you, everyone, and have a good week.